So, good afternoon. I'm happy to be back after almost a week of fairly intense training for those who have been participating in the course. And I now hope to make some outlook in how these techniques you learned, targeting techniques, can be used in a specific biological context. So it's of course not the only context, and I've seen these posters. I actually want to look at them afterwards a little bit, what everyone is doing. But this is one context which I think is a fairly interesting context also for clinical reasons, which and the context matches very nicely to the technical capabilities. So what I'm going to talk about is genomic variability. I'll explain what I mean with that and why we worry about it. And quantitative proteotypes also define this term, and we'll explain what we mean with that. So the general question we're trying to answer, and I think it's a fundamental question in many basic science, but also clinical research areas, is how the genotype, the ensemble of nucleic acid sequences in an organism, for instance in our genome, translates into these phenotypes. So we of course know, even we could also take a picture of this group, that everyone is different. Everyone looks different, has different size, different hair color, there's different other distinguishing features, which are usually referred to as the phenotype. And so for a long time, one could measure these and take photographs and describe them, but one didn't know really where they came from and how they were translated. There is some indication, of course, that genomics or the genotype has some effect on the phenotype, but how exactly it translates is actually an important question, which is largely still unresolved. And what the big difference, of course, to older times is that now we have the ability to sequence through genomic methods a very large number of these genomes, and also with fairly low level of cost and high throughput. So we are in a situation where we have lots of these sequences, we have lots of these phenotypes, we don't really know how they relate to each other. That's why I drew here a black box, because this information somehow goes in this box. It is modulated by the environment, maybe epigenetic information, and then somehow a phenotype emerges. The phenotypes I show here are fairly trivial ones, which are just apparent from a picture, but of course the same arguments would apply to diseases, which have a genetic component. And we'd like to figure out how these things relate to each other. So how has this been researched? It's of course not a new question. It's a very old question, and it has been attempted, and research has been in that direction, has been going on for a long time. Generally, one can group these research approaches into two terms: reverse genetics or forward genetics, and they basically try to start here and walk over here, or to start here and walk backwards. So this reverse genetics might be a case where one would introduce a specific mutation somewhere in a gene, or knock out a gene and simply ask what happens over here. Does anything change in that organism? Unfortunately, oftentimes the organism will die or will be lethal, and so this has been, of course, very widely done, very systematically, and with some success, but it's generally difficult to move from a variant in a genotype over to define a phenotype. The other way around is referred to as forward genetics. One will basically try to group phenotypes into groups and say, what do they have in common? What genetic bases are there that consistently lead that connect a particular phenotype? For instance, this guy has a little hair, and if you had hundreds of those, you could say, what do they have in common? That on the genomic level, that they have relatively little hair, as opposed to others who have more hair on their head. So there's many of these, of course, studies ongoing, and we can ask now, what is the underlying theory? Because like many other fields, for instance in physics, you always supposed to have a theory when you explain some mechanism. And so we can ask, what are the underlying theories or assumptions that would link the genotype to phenotype? And so these are some very ancient concepts. One of them is referred to as the central dogma of biology. So we assume that, and this has been, of course, fairly widely been accepted, and it's not really a theory anymore. It's a basic dogma of molecular biology. And we say there's genes in the DNA. We know them all now. We can locate them. We can determine their sequence. We can say how long they are. These genes are transcribed into RNA, messenger RNA, and then translated into proteins. And somehow or another, we assume that these proteins are determining the phenotype. So there are rules to that, and there's very classical studies which I don't want to cite. And but one of them I think is worth looking into a little more closely, because I think is the prototypical study or situation where the link between genomic variability has been linked to a particular phenotype. And I think the lessons learned from this study have been guiding mostly the thinking of biologists. So this is a case that everyone probably has encountered, maybe not the computer scientists, but everyone who has done biology, and that is the case of sickle cell anemia. So sickle cell anemia is a disease which affects the red blood cells. So rather than being generally in this kind of donut-based shape, they look like a sickle. So they are actually the phenotype that's detectable under a light microscope. So this is a disease which has negative effects. It also has actually some, in heterozygotes, also some positive effects. But that's not really the reason I describe it here. I describe it here because already in the 1940s, this was resolved that a variant in the nucleic acid sequence, a mutation in the genome, will lead to a change in the amino acid sequence of the protein. So hemoglobin is the protein that causes this disease, a variant in hemoglobin. Hemoglobin consists of 2 alpha and 2 beta chains, and one single amino acid in sickle cell anemia is changed in one of the chains. So a single, basically, amino acid exchange goes from a wild type, normal protein, to mutant protein. This was found. It was found that then this changes the structure of the hemoglobin. It changes the biophysics of this hemoglobin by changing the oxygen affinity, the ability of hemoglobin to carry oxygen around the body. And for reasons which are actually probably mechanistically still not very clear, this mutant form causes various forms of the cell which carries hemoglobin, and that's the red blood cell. So at some level, this is a very is extremely appealing situation, because we could sequence one particular nucleotide and say this person is healthy or has this phenotype, and it's a very strong association between the mutation and the phenotype. So this was kind of the guiding light, and many people then thought that this is so rather simple and direct relationship for any phenotype. And this is also the expectation that came out from the Human Genome Project. So this is a picture from Bill Clinton, Tony Blair. They're both defunct now. I mean, they're still alive, but not active anymore in terms of politics anyway, at least not politics in the front line. And they were announcing a monumental event in the history of natural science. And that is the completion of a draft of the human genome. So this was in 2000. And out of this announcement came a press release, so this is generic terms, but then there was also specific deliverables, so to speak, that were expected to be derived from this genome sequence. So what they wrote here, this is a text from the original press release, they wrote that now that we know the sequence of a human, the nucleic acid sequence of a human, we could read out of this nucleic acid sequence information that is relevant to alert patients that they are at risk for certain diseases, predict the course of disease, precisely diagnose disease and ensure the most effective treatment, and develop new treatments at the molecular level. So these are, of course, pretty ambitious predictions. And now with the benefit of about 15 years of research and hindsight, we can say that actually most of these expectations or predictions have not been reached. I think there has been fairly reasonable advances here, that patients based on nucleic acid sequences can be alerted to that they are at risk for some disease or some other phenotype. But certainly this part here has been much more difficult than anticipated. So to translate this press release in some common terms, we expect from genomics, or Clinton and Blair expected from genomics at the time, that risk factors of disease could be identified by reading through the sequence, feeding the sequence in a computer, and then out comes a prediction. That prognostic and diagnostic biomarkers would be apparent, and that mechanism-based medicines would emerge. So again, here I think we're doing quite well, and here we're not doing so well. And I think also the reason we're doing quite well here as a science community is not that we could read this initial nucleic acid sequence, but that technology has progressed over the last decade, that now thousands of such genomic sequences can be created quite quickly and compared. Out of that these comparisons come patterns. So, but unfortunately, it turned out that things are not so simple. And I think this is probably the reason why it has been difficult to generate mechanism-based drugs, to predict biomarkers from genomic sequences. And this is an example here, it's a fairly typical case. So this is a gene that is referred to as a breast cancer gene, BRCA1. And this is a extensively studied gene. It does actually not come out of genomics. It's been discovered before as being associated with breast cancer in women. And if you go to Google, it has millions of entries that have something to say about this gene. So then, of course, if the gene is known, and with genomic sequencing techniques, one could go and sequence a lot of patients that have this breast cancer gene, and it is found in about 1% of women of certain populations. Its presence is thought to increase the risk of developing breast cancer by about a factor of 5, but it is, you may immediately see already, it is not so like in sickle cell anemia, that every time this gene mutation is present, the disease will occur. It occurs not in all patients, not in all individuals that have a mutation in these genes. In about 60 to 85% of women who have a mutation in this gene, they will develop breast cancer during their lifetime, statistically speaking. So this is dangerously close to 50-50 chance. And also many breast cancer cases seen in the clinic do not have a mutation in this gene. So we see the reason I mention this here is it contradicts or it deviates from this very nice coherent picture of sickle cell anemia. If you say, if I have a mutation detected in a particular gene, I can associate it with a phenotype, which is a complex disease. And so here this is not the case. A disease-causing mutation in a particular gene has a risk associated with it. The risk is actually in this case quite high, but it's not absolute, and it also does not mean that if this risk is not present, the person cannot develop the disease. So this contradicts to a large extent the expectation from Clinton and Blair from the Genome Project, in that one could simply read through genomic sequences and make predictions about the outcome or the phenotypic future of an individual. So this, of course, this case raised a lot of public attention through Angelina Jolie, who is exactly in this situation here, and she made the very drastic decision based on this risk factor, which is quite high, but certainly not absolute, is to basically remove the tissues preventively from her body that could potentially be affected by this disease. So this is why this case has been so widely popularized. So now, of course, this is one case, but this is now of a particular gene that has a strong association with a complex disease, in this case breast cancer. But we can now also ask, is there other other diseases, other genes that are involved or associated with a complex disease like cancer. So here is a relative of of breast cancer, ovarian cancer. And this is data that a student in the lab once compiled, Atul Sethi, who was here. And we asked him to basically generate such a plot. And then I'll explain what the plot means. The source data of this plot is genomic sequencing data from the Cancer Genomics Consortium. So this is a consortium which takes patient material, tumor, and also healthy tissue from the same individuals and sequences the complete genome. Of course, you can then compare the genome sequences from the same person and say these mutations or these sequence variants in the same person, between healthy and disease-affected tissue, are somehow associated with the disease. You don't know how. It could just be coincidental, but it could be also causative. And then, of course, you can also do comparisons between the individuals or across the individuals in a cohort. These cohorts were, at the time, this was about 200-300 individuals. Now they're bigger, they're about 1,000 individuals, but the basic picture has not changed. So what Atul did here, he made a plot and said these are mutations that affect certain genes and the number of patients in which these mutations are being found. So this is basically a frequency plot. We simply say how often in a population that was sequenced, how often is a particular gene mutated. That's what this plot is. And we see here an interesting behavior. We see that it's simply an excerpt or a blow-up of this region here. We see that there's very few genes which are quite frequently mutated in this cohort. And we see a very long tail here, which goes actually on, of potentially thousands of genes which have mutations in this patient cohort at a very low frequency. So what did not work here is to say we sequence these cohorts, we compare the sequences, and out comes the disease-causing genes, because there's a lot of variability in there which is occurring at low frequency, and one can either dismiss this as noise, or one can say maybe this information here also, these genes mutated at a rare frequency, are also involved in the causation of this disease. So we can also ask, what are these genes that are occurring with a certain frequency? And so this is what Atul also did. He used the 18 most frequently mutated genes out of 7,000 in this study, and these are the names. I mean, the names are here. These are some of the most frequently mutated genes in this cancer genomic study. And if you look at the names, I mean, they will ring to most people who have read some papers in oncology, ring a bell. BRCA1 actually shows up here. P53, some transcription factors. So basically what came out from these studies is that there's genes are being found which are frequently mutated, which we already knew that they are associated with cancer, and then there is a lot of additional genes out here which are somehow occurring at low frequency. They're certainly not dominant cancer causes, at least not in the same case, in the same sense as sickle cell anemia, that if the gene is mutated, it inevitably leads to disease, but they obviously have also some message to tell to us. So to summarize this part, what I tried to show so far is that there is an important question between the genotype and how this information in here is related to the phenotype, or basically how phenotypes are being determined, in part by genomics and in part by other factors like environmental factors. We are living now in an extremely exciting time as scientists, where this genomic information variability can be measured very cheaply and at a very high throughput and quite reliably. So we basically can uncover the enormous genomic variability in studies even within a single research group with a reasonable amount of cost. And of course, there is also unprecedented levels of phenotypic information available, where people have done big screens of drug resistance, mutagenesis screens, and very large phenotypic screens, for instance, based on imaging. So somehow or another, we still try to compile this or combine these two worlds. We still have a black box. And what I try to show with the case of sickle cell anemia, and the short discussion of cancer-causing or cancer-associated mutations is that that biology is more complicated. Phenotypes are usually complex, and what we learned in school, that like from the case of sickle cell anemia, that a particular mutation associates very strongly with a disease, is a rare case, and normally it's more complicated. So what could we do? And one of the lessons we of course learned from not biology, but from so basically what I'm now discussing is how can we use a different paradigm to explain this connection here than the paradigm that a mutation causes a disease. So we can investigate different kind of scenarios, and here we'll basically look out from biology into social media. So let's assume we would take a coffee break right now, and then we would say in 10 or 15 minutes we reassemble, and we would have a camera, let's say, on the top of the room, and we would take every half a minute, we would take a picture. We would see something like that. We would see that individuals here congregate in groups, especially after the time they have had a week to kind of do business with each other. We would see that these groups probably have different size. We would also see, if we did snapshots, that the groups would change. So for instance, this guy here or this person here might move a minute later over to that group, and we would learn something if we observed the groups over time in many different contexts. We would learn something about who has affinity to the other, who talks with whom, who communicates with whom. And this is basically we want to observe the individual in its context. This would be done here by this very simplistic scenario of arranging people in a room and doing photographs. But of course, this is exactly what Google does, that's what Facebook does, that's what Amazon does. When they track you and your behavior, they collect this information, they will make statements about you and your association, and from the associations make statements about you. So in the world of proteins, we believe that this is also the case. So in the case of sickle cell anemia, we observed a protein as a factor that causes a phenotype. And we now learned from, I think, particularly from these cancer genomic studies, but also other such studies, that this is a rare exception. And we would assume now, at least it's a working hypothesis, that proteins also, as causative agents of phenotypes, also act in cooperation with each other in modules, complexes, pathways, and that it is a module as a whole and not an individual protein. So the protein in context would cause a phenotype, or at least be involved in a phenotype. So this is data from Matthias Gsteiger and Marco, who was a postdoc here, and they tested initially this assumption. So what they did, and what Marco did mostly, is he identified interactions around specifically functionally relevant proteins, protein kinases. But this would probably also true for other types of proteins and enzymes as well. So he affinity tagged protein kinases. They're also shown in this graph here in the center as this octagon. And then he expressed this tagged protein in a cell and asked what proteins associate with these proteins. This is a fairly robust workflow, and so he did this for a large number of kinases. So this is what he found. So let's first describe what the graph shows. The graph shows here always a protein kinase. This is one, they are these octagons. We show other proteins which have either triangular or rectangular shape, and then we see a number of edges. Edges mean that the proteins physically interact with each other. At least what we conclude from this data. So what we see is an interesting picture emerging. We see that most of these kinases have a number of proteins associated with each other. This is certainly not surprising. But then these associated proteins are color-coded, and they're either color-coded because they have a disease-associated connotation in OMIM, which is a disease database, or a cancer association based on COSMIC, which is also a database. So we're not saying at all that these proteins here in this particular cell type are involved in cancer. We're simply saying that proteins which have a connotation with generally disease or generally with cancer are painted onto this interaction map. And the interesting thing is, which supports the notion that I tried to bring up before, that we need to talk about modules and interact the proteins in context, would be that very frequently we see that the kinase itself plus the associated proteins here, the blue guys, have a connotation to disease. So this is the case here. This is for a disease called retinitis pigmentosa. Another one, ALL, scleroderma pigmentosum, and so on. So what we conclude here is that if we talk about the modular biology, that this organization is actually important and not the individual protein, then of course we can use these modules to integrate seemingly different mutations which may be associated with the disease in a functional unit, assuming that this module actually has a functional consequence for the emergence of this disease. So to summarize or finish this part, now I try to explain first how we try to get between phenotype and genotype based on in a molecular biology paradigm. I described how we run into trouble there by associating the inability to associate a complex phenotype usually to one particular gene or protein. And then I introduced the notion that we think about measuring the proteins in their context. And so we define this context as the proteotype and as the acute state of a proteome in a cell, the components and their organization, basically a snapshot how these proteins are present and organized in a cell. And we would predict that the genotype information, genotypic information and variability would be translated into this prototype, and the prototype as a whole, as in contrast to a single protein usually, would determine this phenotype. So these might be modules, might be pathways, or different pathways acting in cooperation. So now, of course, the question is, how can we explore that? It is not currently a plausible suggestion to take a mass spectrometer and to put a proteome into this mass spectrometer, and will tell us the organization of the proteins. So we cannot measure this very effectively. We can measure, as Marco did, and Matthias Gsteiger is continuing to do here, some proteins quite effectively, but it's a lot of work and it's slow. So the only way we currently know is how to make these associations between proteins cooperating in some function is by correlation or association. So we basically make a statistical argument. We would love to measure this directly, but we can't. And so we have to make inference. And so correlation basically, as everyone understands, is that we have variables and we plot them under many observation conditions against each other, and they either behave rather similarly. We talk about a good correlation. So that means if under one condition this goes up on the other, or they have poor correlation, we basically have a cloud, and then there's no information in there. But we assume that if we can generate such correlation analysis, correlation plots, or correlation coefficients, then we would be able to measure association. So if you have a protein and you have 1,000 patients and 1,000 controls, and every time the protein or two proteins do exactly the same when in each patient and each control, then you would say it is not randomly, that is not a random reason that they do the same. There must be some connection, functional, physical, or whatever, but it would show that they have something to do with each other, which is exactly the argument that Amazon, for instance, makes when you buy a book and you and you like this book, and then they will tell you, if you like this book, you might also like the other book, and they do it exactly based on this argument that 1,000 or 1,000,000 other people who bought book A also likely bought book B. Without knowing you, without knowing the customer, without knowing the reason, it's simply by association. Okay, so if we believe in that, then from the measurement point of view, which is really what concerns us here, we would need to be able to have a technology which generates very reliable measurements of proteins, ideally all of them, across many conditions. So only if we have the same protein or the same events, in our case proteins, measured over many conditions, can we generate this correlation or association study. So it is data-driven, and we need good data. And you learned now throughout this week that we have various methods to generate such data sets. And the methods we learned for proteomics is the MRM or SRM, PRM, SWOT, or general DIA. So these are methods which are not perfect yet. I mean, they'll probably never be perfect, but they've made a lot of progress towards the creation of data sets like that, where dozens to hundreds of conditions can be used to quantify proteins. And this matrix is the basis for these association studies. So what are the conditions? This could be clinical cohort, could be time courses, dosage courses, genetic reference strains, and so on. So now, if we accept this view, what I basically tried to explain so far is that to relate the genotype to phenotype, we need a new paradigm. This paradigm are modules, is modular or network-based. We define this prototype, the associations of proteins with each other in the cell or the tissue, and we would not be able to measure this directly. We infer it, then we have described the techniques, or we learned the techniques that allow us to do these inferences. So if we want to accept this view, then we make some predictions which are testable. And in the following, I make 3 predictions out of this kind of scenario, and then I show some data from results from our lab. Actually, some of the people who generated this data are here, and then we show that actually what happens. So one prediction is that the genotype controls the expression of functional protein modules. Second one would be, so this is the connection from here to here. If we of course assume that somehow the genotype determines the expression of a protein, but we would now make the added leap to say, if the cellular proteome, this proteotype, is modular, we would expect that genetics determines also affect specific modules in their quantitative expression. I'm not talking about sequence variants. I'm talking about quantitative expression levels encoded by the genome that determine the presence or absence of a functional module that then relates to phenotype. Second prediction would be that the functional modules, the ones under here, would integrate genotypic variation and determine and relate to the phenotype. That we can make correlations between the proteotype measurements over a number of conditions and relate them to measurable phenotypes. So that this is not a random association. We can learn from this association which factors are determinant for the phenotype. And the third prediction is that the prototype is modulated to various extents by genetic and environmental factors. So it is not a direct run from genome which is somehow hardwired into the proteome, into a phenotype, but that this prototype is modulated. So these are 3 very important points. And for each one, the members of the laboratory have made experiments and studies, and now I'm summarizing these fairly briefly to show to what extent these expectations or predictions that arise out of this view of a modular biology, whether they actually hold up. So the first prediction is the genotype controls the expression of functional protein modules. So this is work that was done by Paola Picotti when she was a postdoc in our group, and the experimental design was that we would use protein quantitative trait analysis in a genetic reference strain compendium. And the experimental approach was to use targeted mass spectrometry, specifically SRM, to quantify components of protein complexes that have relevance metabolic system in yeast. So now I explain these terms up here, and then I show what came out. So the terms are protein quantitative trait locus is abbreviated as PQTL, is basically an association of the level of a protein that is measurable in a cell to a particular locus that determines its abundance. So we assume that if we have proteins in the cell, that the dogma, central dogma says the flow of information goes from the DNA to the transcript to the protein. But that central dogma makes no statement about how much. It makes no quantitative statement. It just says stuff flows. And what we, with this QTL analysis, we make now a quantitative statement. We say we want to find loci that determine the abundance of a particular protein. And that's, of course, what we measure. With targeted mass spectrometry, we measure the abundance of a protein under various conditions. That's what we're doing here. So then I need to introduce the term genetic reference strains, and this is the picture that explains that term. So these are two strains, a laboratory strain and a wild-type strain of yeast that were crossed. And we all know from basic biology, again, maybe with the exception of computer scientists, we know that if these two parents breed, that the genomes, the respective genomes, are shuffled around by meiotic recombination. So basically, the genome that is the one parent and the genome that is the other parent get mixed up to some extent. So if one does this with these strains, one through work from Rachel Brehm, and Leonid Krugliak, who actually generated these strains, then through ingenious breeding, which I don't want to go into, they basically generated in the range of about 100 strains, segregants or progeny strains, that all are, each cell in one of these strains is identical to the other, so they're clones, but between each strain, the cells have a different genome. But the genome is not completely different like one had in wild-type strains, but the genome is structured. That we know it consists of various degrees of various segments and proportions of the two parental genomes. So if these genomes are mapped out, one can generate maps of segments which are either from the one parent or from the other. And if one then measures proteins and protein abundances over these strains, one can make a correlation or association and say whenever let's say the blue gene, the blue allele leads to a high level of expression of the protein, and on a particular locus, and the red allele leads to low level of expression at this particular locus, then one can make an association and say whenever the blue gene was present, the blue allele, then the expression was high. Or go the other way, we can measure the protein abundance and say whenever it was high, does it associate with an allele or a locus wherever the one allele was present, it is always high, and the other way around is always low. So this is what was done here. So Paula generated initially shotgun data to select on selected strains to find proteins which are variable. This association, of course, only works if the proteins actually differ between strains. If everything is exactly the same, there's no information in there. And then she mapped about 2,500 proteins and then selected out of those highly variable proteins that are also functionally related to metabolism. So this was done by SRM. We didn't have a whole lot of channels, and so she took highly variable proteins, projected them on a metabolic map, and then added some other proteins from this metabolic map which were not necessarily shown to be coming out of the initial screen. So basically the upshot is she selected about 50 proteins that would cover important parts of the metabolism and have shown to be somewhat variable across these strains. And then all of these 50 proteins she then quantified by SRM in each one of these strains. So this is a data matrix that came out. So this is something you have also seen now in the throughout the week. So these would be the strains, these are the proteins, and it led to an almost complete data matrix, which of course is nice. So now what does this matrix tell us? It told us, so now we can do association studies and basically try to find the loci that determine the abundance of a particular protein. And so there were actually, out of 50 proteins, we found, and this was done, the computational analysis was done by Andreas Bayer and Mathieu Siza. Complicated French name. But he works in Andreas Bayer's lab. Mathieu Clément Siza. And so out of 50 proteins measured, he found 30 loci that determined this abundance. So this is not super remarkable, but when it became very interesting and really fascinating is when Mathieu and Andreas generated this network out of this data. So what does this network show? This network shows that the yellow dots are proteins that were quantified by Paula in this study. So they were quantified across these strains. The loci that control the abundance of these proteins are shown as triangles, and the edges means that there's a connection. So the way we read that is that this gene here, this locus, determines the abundance of this protein and determines the abundance of this protein. Or the other way around, we would say we have this protein, and the abundance of this protein is controlled in ways mechanistically we don't know by this gene locus. So now this is a fascinating graph because it shows that the loci controlling the protein abundances and the proteins that are being expressed form a very intricate, very complicated network. So this is only admittedly a small number of proteins, about 50. And now we're actually trying to do this on a larger scale. But it is already interesting to see that there is not a one-to-one association. It is most proteins are controlled in some way by genetically and by their expression. This locus here, for instance, controls the abundance of several genes. And we have cases where a protein is controlled by different loci. So this is an interesting case here which goes towards the modularity. These are proteins which are known to interact, these 5, to form a functional complex that carries out the metabolic function. So one might assume that if the cell develops a mechanism to control the expression of a function, and this function is carried out by multiple proteins binding together, forming basically a module, that this might be done by having one locus determining the abundance. That's not the case, at least in this limited network, and we'll see whether this pans out also for larger numbers of proteins and their loci. We see that every single component, with the exception of these 2, but on the others, every single component of this complex has its own control mechanism that controls the abundance. This is, I think, is a remarkable finding because it shows that overall the network of genetic factors that control the abundance of proteins, including the abundance of functional protein modules, is very complex and is certainly much more complex than saying this gene is expressed and some factor controls its abundance. So this, I think, is an interesting finding, and I think we're certainly very keen to explore this further and see to what extent this can be generalized. But what the main take-home message would be that first of all, targeted MS has the ability to actually support such association studies because the data are qualitatively good enough. With SRM, of course, with relatively small numbers of proteins. With DIA methods, much larger numbers of proteins. And then we learned some biology, and so we also learned how genetic variability affects the metabolic network, and that there is actually a fairly intricate or complex network. Okay, so the second prediction we want to make is that functional modules or pathways integrate genotypic variation, and that this proteotype in turn determines a phenotype. So this is actually work that is being done or has been done by Tianan Guo, and so I'm now very briefly summarizing this particular study that was aimed to test this prediction. So the experimental design is to take a cell compendium, and we took, for reasons which will become apparent very briefly, the NCI60 cell line compendium. We quantified, or Tianan quantified, proteins by swath mass spectrometry in each one of the cell lines. Then it used available phenotypic information to correlate the proteotype map here with the phenotypic map from available through the community. So what are these NCI60 cells? These are 60 cell lines that are supported and made available by the NCI, National Cancer Institute of the US. So they're structured in a way that various tissue modalities are represented, so breast and lung, and you can read it yourself, are represented by a number of cell lines. Not a very large number, and one could certainly argue that this number is very, very small compared to the complexity of these diseases. But nevertheless, that's what the NCI makes available. On the average, maybe half a dozen cell lines derived from one particular tissue, and these cell lines are not just made available as cell lines, but they're made available as cell lines plus a huge amount of information, so which has been accumulated over time. So this information would be genomic information, like translocations, methylation maps, expression, transcript information, but also, and that's important for the discussion here, phenotypic information, in that each one of these cells was tested against a very large number of compounds by the Broad Institute, and they determined IC50 values for a large number of compounds, including many drugs that are used in the clinic for cancer therapy. So this is basically our phenotypic readout. So what Tianan attempted to do, or did, was to take these cells, extract proteins, generate swath maps, identify and quantify as many proteins as it could from these cell lines, and then basically define this prototype, and then correlate this with the drug response or sensitivity or resistance that has been noted by these cell lines in the past. This is the data that came out, remarkable data set, I think. So these 60 cell lines were run in duplicates, and Tianan thinks he identifies and quantifies about 5,000 proteins across all these cell lines. So we can then start. So this is just to show that the abundances are actually the abundance distribution is quite tight, and the data are of good quality. So now I skipped that for reason of time, and now we can take this drug resistance or drug sensitivity information, and we can relate it to the prototype, the proteome measured in these cells at the instance when they were lysed. And what we try to do here is to say, is there protein patterns that correlate with the sensitivity or resistance to a particular drug? And of course, once we have these patterns, we can then also say which proteins are responsible to mediate this phenotype. But that comes later. So here is a graph that illustrates some of this data. This is one particular drug, MEK inhibitor, which it's really not particularly relevant here for this why I show this particular one and not any of the other hundreds of such graphs. And what we see here is a kind of a phylogenetic tree-like structure. And so each dot here is one of the NCI60 cell lines. The cell, and where the cell lines are in this graph, whether they're close together or where they are, indicates how close they are to each other at the level of the proteome map. So this cell line here would have a quite different proteome than this cell line here, because it's far away in the graph and it's far away in the proteome and the proteotype map. So we can see here that there is proteins clustered, cell lines clustered together, so they are quite similar in their proteome, and some are far apart. So now Tiannan painted these cell lines with a certain color, and the colors we see is cold colors or hot colors, and the hot colors are resistant cell lines, and the cold colors are cell lines that die when the drug is added. And so we can see that there are resistant cells, but there's not one type of resistant cell according to the proteome. So from the phenotypic measurement, these cells are just resistant, and so they do not die when the cell is exposed to this drug. However, when we look at the molecular phenotype, the proteome, there is several ways how these cells can become resistant, based on if we assume that the proteome is mediating actually the resistance. So we have one cluster here, we have one cluster here, and we have one cluster here. So we would predict that there is 3 different mechanisms mediated by protein patterns to make these drugs resistant, these cells resistant to this particular drug. And so we can, of course, since this is a classifier, we can ask which proteins make these cells go into that cluster and not in that cluster. So this is the next graph. These are the abundance levels of particular protein across these cell lines in the various clusters, so the various resistance cluster 1, cluster 2, cluster 3. And these cells, these proteins have a particular names, which we don't need to belabor here, but they have actually interesting names, and they're plausible why they might be involved in the generation of resistant, a phenotypic effect. So these are transporters, these are metabolic enzymes, or that may metabolize the drug. There's another transporter, or a regulatory protein, a kinase. So what we basically assume here is that once you have the cells present that are inherently can be in different states based on the proteome. Some proteomes lead to a particular phenotype, which is measured here as resistance to a particular drug. And what the data show is that there is different proteins that can contribute based on associations, not mechanistically. The mechanistic investigation will come now, where we would maybe say, okay, we want to test whether a particular membrane transporter actually is able to, for instance, kick the drug out. This would be a follow-up mechanistic study, but this is simply by association. We can identify proteins which statistically associate with the phenotype of making the cells resistant. So this is not a unique, this particular MEK kinase inhibitor is not a unique case. In fact, it's a common case in the sense that we have now a variety of these graphs. They're all for a different inhibitor, and we see that in every case here where the graph was generated, there was more than one cluster of cells that are resistant, that are from the proteome not close to each other. So we would say it is a fairly general statement to assume that cells can be resistant to drugs by different molecular mechanisms. These molecular mechanisms are apparent at the level of the proteome, although we don't understand them explicitly. They're simply being derived by association, but they are different from each other. And this is, of course, important information, for instance, in oncology treatment. Okay, to summarize this part, what I try to test here with Tiannan's data is that functional modules or pathways integrate genotypic variation and in turn determine the phenotype. So we can see that massively parallel targeting generates big data sets in the arena of proteomics through the techniques you got to know this week, that we can correlate these prototypes with phenotypic information and identify mechanistically important proteins, which could be tested further on how actually mechanistically they relate to the phenotype. But so far it's by association, but it is fairly strong evidence. And then we think that this proteotype provide complementary information to transcript profiles. I don't have time to discuss this, but it's not that we just duplicate with another technique what everyone already knew from transcript measurements. So now, I'll quickly, I hope, go through this third prediction and say that the proteotype is modulated to various extents by genetic or environmental factors. So what I would like to demonstrate with this last vignette is to say, yes, we can have a genetic component that determines the phenotype and actually the proteome, but it is by no means the only factor. There's other factors which act on the plasticity of this proteotype, and we would like to know which effect is genetic and which effect is from environmental factors, for instance, age, living conditions, and so on. So this is work from Yansheng Lu, who was a postdoc in the group, and he is right now not here, he's in China. So he undertook to use twins, a twin cohort, to ask these questions that I posed at the very beginning. So more specifically, he took blood plasma from twins and the measured proteins in this blood plasma. So why twins? Because twins have an interesting characteristic that monozygotic twins are thought to be genetically identical. And dizygotic twins are not identical, but obviously related. So all the monozygotic twins, they have shared genes. So if there is a genetic determinant that determines, for instance, the abundance of a protein, that determinant should be present in both twins. Whereas it is not the case in dizygotic twins, which differ in their genotype, actually to the same extent as the strains in genetic reference strains differ, because they're also derived from two parents. Okay, so this is the general idea of this type of study. We would plot, as in a correlation plot, the expression of each one of the detect proteins across monozygotic and dizygotic twins and twin pairs, and determine a correlation factor. If a protein expression, if the expression of one protein is very strongly determined genetically, and then they would be on one line, because they're genetically identical, so they would presumably have the protein would have the same abundance in both twins. And that's not necessarily true here, because they are not genetically identical. But if a protein in the monozygotic twins is fairly far spread out, this would mean that in addition to genetics, or in the absence of genetics, there's other factors that determine the abundance of the protein. So out of that is this formalism that has been developed by population geneticists for a long time ago, basically using these correlation coefficients from population measurements. We can calculate whether, if this is a particular phenotype, maybe the expression of a protein, we can divide up the contributions to the variability observed in that phenotype over the population into genetic contribution, environmental contribution, or other longitudinal effects or non-biologicals, which basically is unexplained. So he did that. He took these twins. These were 36 monozygotic twin pairs, 22 dizygotic twin pairs, and he measured them at 2 times in the life. So these were samples we obtained from Tim Spector, who maintains a twin database in the UK. He's very generously made accessible these samples to us. So this is a sample set of 232 measurements, because he did tandem measurements of these individuals. He did this by swath MS. This is what came out. These are the twins. These are the proteins. He quantified persistently about 340 proteins. This may not seem a very high number for for you after this course, but it is actually a remarkable number for plasma because we did not want to fractionate the plasma, nor did we want to deplete it. We wanted to just take the plasma, digest it, to have a minimal number of artifactual potential variability introduced experimentally. Okay, anyway, that's the data set. And then is this, we could first ask, does this data set actually have a signal? So we can ask here, these are the twin samples, and these are technical repeats or repeats of the whole process. And this is the coefficient of variation. So what this plot basically says, that technical or biological repeats have much lower CV than coefficient of variance than the actual sample. So this indicates that there is biological signal to be obtained in there, because if these were all curves all overlapped, we would have no biological variation to measure. It would simply be whatever variation we measure is technical, which is not the case. So then he can proportion to these 342 proteins the source of variation, the root of variation, basically the reason why they vary. So we see these are the proteins on the x-axis here. This is the percentage of variation of contribution of the variance explained. So this is the total variance observed for one protein. And then he proportions this total variance into either genetic components, environmental, or unexplained. So we see here proteins that are largely genetically controlled. Quite strongly, maybe half of the variability observed is genetically controlled, which is kind of remarkable. And the rest is by some other mechanism. We see also proteins which seem to have virtually no genetic control. So they don't really care about the genetic background in their abundance levels, but some have strong longitudinal effects. That means at the older population, the variation was larger than in the younger sample, and we have a whole number of proteins here which have the bluish guys, which have strong component of environmental control. So it is by no means so that every protein behaves in the same way. It appears that every protein has its own kind of life, in the sense that who controls its abundance throughout the duration of a person's life. Some are fairly tightly controlled genetically, others seem to be not so tightly controlled genetically, but have a strong environmental effect, and others seem to change systematically longitudinally. This, incidentally, is an important finding for biomarker studies, because if you were to place a biomarker in here, where mostly the proteins are quite variable and the variability is strongly influenced by environmental effects, you would of course have a hard time to find a marker which separates populations well. So these are just then to show, I think I skip over that, it's just to show that these various mechanisms of control are not randomly spread out over the proteome, but they go with specific functions. For instance, immune response, blood coagulation, so on. I skip this, and I just want to show that generally, which is also an important factor, I think, for clinical studies, is generally speaking the genetic control of abundance of proteins decreases with age. So one could assume that in a newborn, the genetic factors are strongest, and then maybe through somatic mutations or other mechanisms, maybe environmental effects, the genetic control slowly decreases, and the other factors which affect the variability of the blood proteins eventually increase. These effects are not dramatic, but they are statistically highly significant. I also would like to remind you that the time difference of these two measurements is only about half a decade, so it's by no means the whole lifespan of these individuals. So I skip this for reason of time. We then also were able to use the quantitative information to, again, like in the yeast strains, to link the variability of a protein back to a locus that determines it, and I think that I just want to skip over that and come to summary. What I tried to show in this last vignette is In the twin cohort, in genomically structured cohort, we can identify through consistent measurements of the proteome afforded by the methods we discussed this week. We can get high quality data sets which allow us to, based on the genomically structured cohort, determine the source of variability, where it's coming from. Whether proteins are variable, they are variable. We can now not just say they're not. We can not just say they're variable. We can also say they're variable because of genetic or other factors. We see that strong longitudinal effects confound biomarker study. If someone is in biomarker studies, we can identify pQTLs, loci that control the quantitative prototype. Although these were, again, not a large number of proteins, it was like 340 proteins. If one did this with a 10-fold higher number, one would get probably also a very interesting network of loci that control the abundance of specific functions. And similar considerations apply also to PTMs. I mean, in fact, Yan Sheng, together with George Rosenberger, have developed a system to revisit these swath maps of these proteins, these plasma proteins, and find PTMs that are also genetically have various fractions of genetic control. So to put it positively, they found PTMs which are under genetic control in their abundance, which is an interesting fact because many times people say that PTMs are, it's difficult to distinguish which ones are actually biologically meaningful and which ones are not. And of course, the assumption is that if something is genetically controlled, then it is biologically meaningful because it was selected by evolution. So anyway, I'd like to finish here. And what I showed is some data which rely on swath or MRM measurements. Is the group of people who developed the swath techniques here. The kinase interactome data I showed one slide. This was the work mostly of Marco Mariosolo, who works with Matthias Gsteiger. The twin project is really mostly the work of Yansheng Lu, and he works on the data analysis with Alfonso Buil and Manolis, Dedmitsakis in Geneva, and the samples came from Tim Spector. The NCI 60 project is the work of Tianan, and he works with Julio Saez Rodriguez at TBI for data analysis, and the yeast pQTL study was from Paula with Mathieu Clément Cisa. Now I got it right. Now it vanished again. And of course, as you experienced through this week, nothing would happen without software. We particularly acknowledge contributions from the Skyline team, from Lukas Reiter, Manfred Klaassen, and the present and Pedro and the present informaticians or informatics students, Hannes and George, and whereas Hannes just left. We also are very grateful for Olga Witek and Mina, who keep us on track and keep us from making stupid statements, at least statements that are not statistically supported. And it's been extremely valuable so far, the interactions with both the Skyline team and with Olga, because I think they have competences which we don't have in our group to the same extent. So I'd like to stop here and thank you for your attention. So if someone has a question, I'm happy to try to answer it. Yeah, Olga.
For future studies in this field, do you think the main bottleneck currently is the throughput of measurements, or would it be more important to have technologies that cover more proteins? I mean, would it be more important to be able to measure hundreds of samples, or rather 20,000 proteins in every sample?
Yes, so that's actually a good question, and we spend a lot of time discussing this. So in the field of proteomics, there has been, and still is, if you go to meetings like ASMS or some proteomic meetings, there is an enormous pressure to come up with very high numbers of proteins. And so the current standard is like 8 to 10,000 proteins identified from a mammalian sample. we think that, and I showed like the first study from Paula, and I showed this last study from Yansheng, where a relatively small number of proteins measured across many conditions provided extremely interesting biological information that would not be apparent from having only 5 or 10 samples measured, but much deeper. So I personally think, although this is more right now more of a gut feeling, and actually we had just a very interesting discussion along these lines also with Olga Vitek this week, that I personally think that much more throughput-oriented proteomics, where we can do, instead of hundreds of samples, thousands of samples, but maybe at the cost of some of the depth of the proteome, will be very beneficial, provided that we can make these measurements very highly reproducible and quantitatively accurate. So my personal opinion, although I could not cite a very strong reason except these anecdotal cases, and some other anecdotal cases, like Yibo Wu, who sits at the back, has done a similar study to Paula, but in mice, a similarly structured mice cohort, again with very interesting findings. So I think that by having larger numbers, not so deep, is better than very deep and small numbers, more informative anyway. And of course, what we like to do is very large numbers and very deep. Okay, so looks like everyone wants coffee. Thanks for your attention.